Hello friends, welcome back to my channel Medicine of 101. Today we'll talk about physiology of thyroid hormone. So before starting the video, let me tell you, I have already uploaded the video based on the thyroid, hormone productions in the part 1. So how the, iodide things that will be taken in terms of the raw materials, how different kinds of the reactions in terms of the organifications, coupling reactions, ultimately we'll get the productions of the T3 and T4. All these things I have already explained in the part 1 video. Okay, so please first watch the part 1 video, then come to the next one. Okay, then it will be easier for you to understand clearly the whole things. Now see, in the part 1 video I already said you that through the diet, through the diet you will get the what? Iodide. So as let's our recap on very soon. As I said you, through the diet you will get what? Iodide. It will come over within the circulations, and from the circulations to the what? Iodide pump. Is active transport. So after that we will get the iodide pump that will be working over here, and the iodide will get where within these cells. Now whenever we will get within the follicular cells, whenever it will go where within this, the apical surface at that time will get, or we can say the luminal surface, because we will get what? We will get a colloid or the lumen sites over here. To understand from the first video, I already explained you that we will get a lumen over here. We will get a lumen, this type of the thing. From the each cells, we will get the productions of the what? Iodide. And there is a presence of an enzyme that is known as what? Thyroid peroxidase, thyroid peroxidase. So for example, is the thyroid peroxidase enzyme, thyroid peroxidase, and that will convert this iodide into iodine, and we'll get, by the actions of this enzyme, we'll also get the productions of the what? Thyroglobulin protein, or in this follicular cells, thyroglobulin proteins, and these thyroglobulin proteins, they are having the what? Multiple tyrosine amino acids. So for example, the tyrosine amino acids, tyrosines, and I said you try to remember from there that the tyrosine, they will get combined with the what? This iodine. So let's use this color only. So it's the chain, peptide chain, and here we will get what? The tyrosine residues, tyrosines, and this iodine will get bind over here, and they will form what? Different kinds of the things. They can produce MIT, monoiodotyrosine, they can produce DIT, they can produce T3, T4. How it will be produced, I have already explained in the first video. Okay. Now let's talk about some, the drugs, or you can say some, anti-thyroid drugs. I'll just say very briefly. Yet we'll have a different lectures on, the drugs that are acting well in terms of the thyroid gland. But for now, I'll just say it very briefly. Okay. Now say that there are certain kinds of the drugs that can act where within this iodide pump. So let's write down the name of the drugs. Okay, this part. Now see, there are certain kinds of the drugs that act within the iodide pump. So, drugs act on iodide pump. So there are certain kinds of the drugs, they act on the iodide pump. What is the name of the drugs? For example, you can get the thiocyanate, or you can get the perchlorate. These are the what? These are the anions. Now what they will do, they will come over here. See, they will come over here, and after coming over here, they will use this iodide pump for them. They will use this iodide pump for them, and they will go over within the cells. Okay, these drugs, for example, the thiocyanates, or you can say the perchlorate. And another thing they will do what? They will just inhibit this iodine intaking. That means iodine uptaking, that will be inhibited by this drug. This is drug that will act as antagonist over here. So try to understand, this drug will block these portions where the iodine is supposed to be bind, and as a result we will see the iodine that will be repelled back. So we will not get the iodine while within this follicular cells. So whenever we are not getting the iodine, try to understand, definitely the iodine will be also a deficiency of the iodine we will see at this level. So whenever the iodine deficiency at this level, definitely the productions of the T3 and T4, that will be less. So whatever the thing we'll get over here, try to understand, whenever the T3 and T4 is less over here, whenever the T3 and T4 that is less over here, try to understand within the circulations we will get also what? The T3 and T4, that will be also less. Within the circulations we will get T3 and T4 both will be less, and whenever these things will be less, what? the TSH hormone that will be secreted more amount. Why? Whenever it will be less, at first within the hypothalamus, the TRH with the positive feedback, TRH, this hormone that will be secreted, TRH, and from here the signal will come anterior pituitary, and from here we will get the secretions of the what? TSH, thyroid stimulating hormone. So within the circulations we will get the amount of the TSH that will be elevated. And what do we know? That within the follicular cells over here there is the receptors for TSH. So whenever for example, is the receptors for the TSH. So whenever the TSH that will come over here, and that will give the signals. Whatever the signals it will do, at first whenever it will be activated, TSH will be activated, it will give the signal within the TSH receptor that is present within the thyroid follicular cells. Okay, at first it will do the what? It will increase the size. If we see in this way, that it will cause the hypertrophy. It will cause the hypertrophy of thyroid gland. Second things, that means what? It will increase in the size. These things that will help to increase the size of the what? Thyroid gland. Second thing, it will just do also the proliferations of the follicular cell. So hypertrophy of the follicular cells will get first. Then we'll get the proliferations of the follicular cell. So proliferation. So whenever the proliferations we will getting, so number of follicular cells, that will be also increased over here. Okay, and try to remember over here. Along with that, it will also try to elevate the level of what? T3 and T4. T3 and T4. It will try to elevate. But the problem is over here, we are using certain types of the agents, anti-thyroid agents. They are coming over here. They are not helping to take the iodide from the what? From the circulations to the within the follicular cells. So whenever iodine deficiency is over here, definitely T3 and T4 that will be less. So whenever it will be less, TSH automatically it will be elevated. So whenever TSH will be elevated, it will come to its receptor and it will activate it. As a result, we will get the hypertrophy of the thyroid gland. We will get proliferations of the thyroid gland, and we will get also the T3 and T4 that will this TSH will try to elevate. But ultimately, if we see in a practical way, that will not elevate it because iodide pump that is already blocked over here. So this type of thing, whenever you are getting the size and the number of cells that is getting increased, these things are that is known as goiter. We will get swelling of the thyroid gland. Now one thing you can remember over here, if you the thyroid hormone, amount of the thyroid hormone, that is inversely proportional to the weight of the thyroid gland. That means try to understand from here. If we say that thyroid hormone is getting decreased, definitely weight of the thyroid gland that will be elevated, as we are getting over here in terms of the goiter over here. Okay. Now we'll talk about the second groups of the drugs. Okay. Before going to the second group of the drugs, let me tell you, we can use certain types of the also drugs that can increase the amount of. Okay, let's see it in the practical way, then it will be easier for you. Okay. First, we'll talk about the second group of the drugs that will act where within the thyroid peroxidase enzyme. So the drugs that act where within the thyroid peroxidase, thyroid peroxidase enzymes. So drugs, if you write it over here, the drugs that acted where within the iodide pump. I will talk about the drugs act on thyroid peroxidase enzyme. What type of the drugs will get over here? We'll get the thiouracil, we'll get propyl thiouracil, we'll get the carbimazole. Okay, we'll get the what? Propyl thiouracils, we can get the carbimazole. What's their functions? They will come over here and they will inhibit this enzyme. So whenever they are inhibiting this thyroid peroxidase enzyme, so they are inhibiting what? The thyroid peroxidase enzyme. So whenever these functions will be, try to understand, that will be what? Decreased or inhibited at the time, definitely the iodide that will be not able to get converted into iodine. Iodide, it will be still in the anion form, it will not in the I2 form. So whenever we are not getting I2, definitely the MIT, DIT, or you can say T3, T4, that will be less. So definitely we can say ultimately within the circulations, the T3 and T4 that will be less. In the same case, we'll get over here the TSH that will be elevated, and that will come over here and that will activate these receptors, and ultimately we'll get the goiter also over here. So if we use the if we can use the drugs propylthiourasis or carbimazoles over here, that will inhibit the or thyroid peroxidase enzymes. So whenever it is inhibiting the thyroid peroxidase enzyme, definitely the T3 and T4 that will be less within the circulations. Will get the amount of the T3 and T4 that will be less. Ultimately, that amount of the TSH that will be activated, and TSH having the receptors on the follicular cells, the TSH will come over here and it will activate. Ultimately, we will get the hypertrophy of the follicular cells. We will get proliferations of the follicular cells, and that will develop the goiter. Now here we can write in a nutshell that the same thing will get over here, the T3 and T4 that will be less. So whenever it will be less, try to understand from here that definitely we'll see the TSH that will be elevated, and we'll get the goiter also over here. We'll talk about all these things when this type of the drugs we are supposed to use and not. Okay, I just said in generally, I just said in terms of the general introductions, but for example you can consider in terms of the drugs. Now we'll use another kind of the drugs just before, if we just doing any kind of the surgery within the thyroid gland. Just before doing the surgery within the gland, we use one type of the drug that is known as potassium iodide. So if we write it over here, that before surgery, okay, let's write it over here. Before surgery, we use our potassium iodide. Now, why do we use the potassium iodide? Let's try to know about it. Okay. Now, so let's talk about these things. This iodide pump. This iodide pump is, its behavior is a little bit different things. Now, see, whenever the iodine within the circulations, we will get over here within the interstitial space, or you can say within the circulations. Whenever the iodide that will be less, at the time the pump that will be more active. See, let me repeat once again. Whenever the amount of the iodide within the circulations or within the interstitial space that is less, at the time the iodide pump that will be activated more. But whenever the more amount of the iodide we will get over here, that will be less activated or underactivated, or you can say whenever the concentration of what iodide that will be severe, at the time this pump that may be inhibited. So for example, we are getting, okay, let's show this part over here. Where are you? For example, we are getting another iodide pump over here. So whenever we are getting the iodide pump over here, whenever we are using the, before doing the surgery of the thyroid gland, we are using the potassium iodide. So whenever we are using the potassium iodide, from here we will get the much more amount of the iodide. We will get much more amount of the iodide. So whenever we are getting more amount of the iodide, at that time try to understand this pump, as I said you, whenever the amount of the iodine or iodide within the circulations or within the interstitial space, that will be much more. The concentration of the iodide will be more over here. As a result, this pump, that means we can say the iodide pump that will be inhibited. So whenever it will be inhibited, try to understand definitely the iodine that is not going well within these cells. Now the interesting thing is over here, this potassium iodide that will not cause the goiter. The potassium iodide don't cause goiter. Rather it will decrease the size of the thyroid gland. It will decrease the size of the thyroid gland. Size of thyroid gland. We can see in a nutshell we will get the shrinkage of the thyroid gland. So whenever we are getting the shrinkage of thyroid gland, it will be easier for the surgeon to do the surgery. Okay, it will be easier for the surgeon to do the surgery. Okay, I am repeating once again. Before doing the surgery, you can use the potassium iodide. Whenever we are using the potassium iodide, the concentrations of the iodide that will be much more. So whenever the concentration will be more, at more concentrations, okay, whenever the severe concentration will get over here, at more concentrations, the iodide pump that will be inhibited. So whenever it is inhibited, definitely the iodide that will be not able to go from here to here. So whenever it is not going in these cases, we can think about the goiter formations. But the most important thing regarding this drug, that it will not lead to formations of the goiter. It will decrease the size of the thyroid gland. Okay, so you can remember in terms of the surgery, if we use potassium iodide, that will decrease, and that will just cause the shrinkage of the thyroid gland. So it will be easier for the surgeon to do the surgery. Okay. Now we'll talk about, okay, So in these cases, we'll get what? Size of the thyroid gland that will be decreased. Along with that, remember, you can remember functions of the thyroid gland that will be remembered. That can you remember? We will also get the vascularity within the thyroid gland that will also decrease. So when you are using the potassium iodide, this potassium iodide that will decrease the size of the what? Thyroid gland. That will also decrease the function of the thyroid gland. That will also do the less vascularizations within the thyroid gland. So that's why before surgery we can use the potassium iodide. Okay. Now we'll talk about these things as we continue in the first part. Now, what did I say up to the last time, that from the follicular cells, thyroid follicular cells, what are the thing we'll get from here? We'll get the productions of what? T3 and T4, right? We'll get T3 and T4. Now, whenever they will come within the circulations, they will get bind with the certain kinds of the protein, and those proteins that will be secreted by the liver. Now, whatever the proteins will get over here, try to understand. From here, we'll get the productions of the different kinds of the protein. One of the protein that is known as thyroid, binding globulin. Thyroid binding globulin. I am writing it over here. TBG stands for thyroid binding globulin. Thyroid binding globulin. Okay. Now, this T3 and T4, they will come over here and they will get bind with this protein. See, but if we say that T4 is having the more affinity with these things, but T4 is having more affinity, so they will get bind. They will get bound what? They will get bound tightly within the thyroid binding globulin protein. Okay. Along with that, we will get also the productions of other kinds of the protein from the liver. We will get also the productions of the prealbumin, thyroid. Okay, let's write it over here. We will get the prealbumin protein, where we will also get the binding of thyroid in terms of the T3 and T4, but less amount of the T3 and T4 that will get bind within the prealbumin part. So T3 less amount and T4, for example, T3 we are getting T4. Okay, another kind of we will get over here. Okay, let's talk about those things. Yes. So we are getting our thyroid binding prealbumin. Then another type of the protein we will get over here, that is known as thyroid, thyroid binding albumin. Albumin. Here, the very less amount of the T3 and T4 they will get bind. But see, as I said you that T4 will get bound where? Tightly along with this thyroid binding globulin proteins. Okay, it is having the more affinity for this thing. But if we see the biologically more active form, what? The biologically more active form, that is the T3. T4 is not biologically active. So whenever you are getting this type of thing, for example, if I show it over here, okay, let's try to draw the structures for this thing. The tyrosine residues molecules in terms of the amino acids, Okay. Okay, let's use the different these things for you. You can just attaching this to tyrosine molecules. And then third and fifth positions is the third positions where we are getting the binding of the what? Iodine molecules. In the same way, we will get also over here the binding of what? Thyroid, the iodine molecules. We will get over here, we will get over here. Now what is the T4? It's what is the T4? T4 is biologically what? T4 is biologically inactive. Now there are certain kinds of the enzyme that is present within the circulations that can convert the T4 into T3. This is the T4, and there are certain kinds of the enzymes that can what? That can convert the T4 into T3. That is in the biologically active form. So what is the name of the enzyme? Okay, let's write it over here. The enzyme that is known as 5' mono diiodinase. 5' mono diiodinase. Now, whenever we are using the 5' mono diiodinase, at the time, what type of thing we'll see over here? We'll get the removal of what? We'll get the removal of on iodine group from here. We'll get the removal of on iodine group from here. So whenever we'll get, it's so over here. Okay, you can consider this the third group, this is the fifth. So yes, Yes, I just did in the same way. We'll take this is the 5 prime, so the 5 is the 3 prime, for example. Okay, guys. Now see, whenever you're using the 5 prime monodiodinase, when you are using the enzymes 5 prime monodiodinase, it will just remove the iodine group from the first tyrosine molecules, for example, from here. So whenever it will be removed from here, whatever the thing will get over here, understand from this figure. If we get further, okay, let's okay. Now see, whenever removing the iodide group from this position, so definitely the iodine that will be removed from these 5 positions, 5th positions of the carbon, and we will get only the iodine over here, we'll get the iodine over here, we'll get the iodine over here. Now this type of the thing that is now, right now we can say what is the T3. Is the biologically what? Biologically active form, biologically active form. Okay, biological is the biological active form over here. Now sometimes we see, for example, there is a certain types of the enzyme that can also, if we say that this iodide molecule that can be also removed. But we want the iodide molecule removal from the fast tyrosine molecules. So this will be biologically active. But if we get the removal of the iodide group from these positions, that will be known as or reverse T3. That will be known as reverse T3. But that is not biologically active. We have to remove the iodide molecule from this 5th prime positions from here. Okay, so let's see it once again. So as I said, the T4 that is biologically inactive. So whenever we get the T4 in the circulations, okay, so T4, there are certain kinds of enzymes that can remove one iodide group, iodine group from the tyrosine molecules. For example, from here in the 5th prime positions. Now for that we are having the certain types of the enzymes that is known as what? 5 prime monodiodinase. So whenever it will be activated, it will remove the iodine group from here. As a result, this will get the biologically active form. But if it removes from the iodine from these positions, not from the first tyrosine, that will be a reverse T3. That is not biologically active. Okay. Now if we say in terms of the half-life of the T2 and half-life of the T3 and T4, try to understand from here. If we say the half-life for T4, that is around 6 days. So if you inject once the T4 within the so at that time if we inject the T4 at that time, it is having the chances for be within the circulation for longer durations. But what about the half-life of the T3? See, the half-life of T3 around 1 day. So it will come and it is having what? The rapid actions. It is having the rapid actions. Okay. So it is having the half-life for T3 that is 1 day, and the half-life for the T4 that is around 6 days. So this type of the thing we'll get in terms of the what? Actions. That means in terms of the durations within the circulation. Now we'll talk about certain kinds of the conditions. For example, as we know that during pregnancy, try to understand during pregnancy, let's write it over here. Then I think it will be easier for you. Let's try to write it over here. So during pregnancy, what we'll get over here? During pregnancy, the liver that will secrete more amount of the what? During pregnancy, the liver that will secrete more amount of the thyroid binding globulin protein. So whenever it is really, whenever it is secreting more amount of the thyroid binding globulin protein, as a result, we will get that more amount of the T3 and T4 they will get bound along with this thyroid binding globulin protein. So within the free form, if we say, try to understand, if we say that T3 and T4, 99%, try to understand from here, 99% of T3 and T4, they are bound with protein, and we will get around 1% T3 and T4 if we say they are in the free form. That means this free form they are biologically active. So see, 99% of the T3 and T4 they are get bound along with the thyroid binding globulin proteins and the other proteins that is secreted by the liver. And around 1% of the T3 and T4 that is in the free form within the circulations, that is the biologically active form. So as I said it over here, during pregnancy we will get the amount of the thyroid binding globulin protein that will be elevated. As a result, the more amount of the T3 and T4 they will get bound along with the thyroid binding globulin. So within the circulations, the free form of the T3 and T4 that will be less. So whenever within circulation spot these things, try to understand the T3 and T4 that will be less for increasing the value of thyroid binding globulin during pregnancy. So whenever it will be less, definitely the amount of the TSH that will be elevated. elevated. So during pregnancy we will get the thyroid binding globulin protein that will be secreted more by the liver. So here it will be more secreted by the liver. As a result, the T3 and T4 they will get bound more in amount along with the thyroid binding globulin protein. So within the circulations we will get the free T3 and T4 that will be less. So whenever the free T3 and T4 that will be less within the circulations at the time, definitely as we know that amount of the TSH that will be elevated. So that will be elevated over here. Other conditions we can get in terms of the liver cirrhosis, we can get certain terms of the problems. During liver cirrhosis, it will be secreted less amount of the what? Less amount of proteins in terms of the thyroid binding globulin proteins, in terms of the thyroid binding, pre-albumin, thyroid binding, albumin. So whenever it will produce less amount of this, binding proteins as a result. So more amount of the free, more amount of the what? T3 and T4 that will be in the free form. So more amount of the T3 and T4 as a result, the TSH level that will be, decreased. The TSH level that will be decreased over here. So during pregnancy, as I said, during pregnancy, the liver that will secrete more amount of the thyroid binding globulin protein. So as a result, more amount of the T3 and T4 they will get bound along with the thyroid binding globulin protein. So if we see within the circulations, the free form of the T3 and T4 that will be decreased. As a result, the TSH level that will be elevated. Okay, and if we see in terms of the liver cirrhosis, the liver that will secrete less amount of the what? Thyroid binding globulin proteins. Okay, so whenever it will produce less amount, try to understand. During that time, we'll say that the free form of the T3 and T4 in the circulation will be more. As a result, the TSH by the negative feedback new positions will get, process will get the T3 and TSH that will be less in circulation. So this was all about, I think, in terms of physiology of thyroid hormone part 2. In the next part of the video, we'll talk about how the pituitary and the hypothalamus they are controlling, how the pituitary and hypothalamus they are controlling, this whole process, how they regulate the secretions and different kinds of the molecular mechanisms within the cells. We'll talk about it in the next videos. So if you think this video is helpful for you, please do like and subscribe my channels, and see you in the next video, and keep watching and please support.